Hello, everybody. This is Julian speaking. I hope you can hear me well. We are excited to start the session. We will wait another minute for everybody to come in, and then 12:30. Let's wait a little bit, then we will start the presentation. All right. Hello, everybody. I'm super excited to kick off the second day of the Selling Partner Conference. With me, I have Guillaume here, and today we will have a session on our technologies for single cell omics analysis, and we will start with a few slides as an introduction to our portfolio, and then Guillaume and me will show you live product demonstrations from our labs and finish with a Q&A session. And my name is Julian. I'm the CSO at CYTENA, and as I said, I'm super excited to guide you through this presentation.
Hi, good morning, everyone. I'm Guillaume Tournier. I'm the co-founder of cellenONE, one of the latest company to join the selling family. And we look forward to your question and then comment at the end. So don't hesitate to ask questions during the sessions.
All right, so let's get started. For the presentation, I will switch off my camera and we'll come back later when we have the Q&A session. Just a few housekeeping rules. We are working with GoTo, so on the right side you have this panel which you can expand with this red arrow, which allows you to access, for example, the questions panel, where you can post questions throughout the entire session. We will then address them at the end of the presentation after the instrument demos. So please feel free to add questions here whenever they come up. Please also note that throughout the entire session you are muted, so you need to use this question panel to submit your question. And I think with that we are good to go. Let me start with a brief overview of the content we want to go through today. I want to start with showing you the latest additions to our portfolio of instruments for single cell omics analysis. We will then talk briefly about some selected application examples, but then the main highlight of this session is that we show you instrument demonstrations from our labs in Lyon, in Freiburg, and in Stuttgart. I have to say that they are not exactly live. We recorded them this week for this conference session to make sure everything runs smoothly. And then we have a Q&A session where both Guillaume and me will be there for you to address any questions around our instruments or around setting up single cell omics workflows. let me start with two slides highlighting some of the latest additions to our portfolio. What you can see here is the I.DOT Mini, which is a very compact instrument, which is based on our proven I.DOT technology for low volume reagent dispensing. And the I.DOT Mini is especially useful when it comes to dispensing PCR master mixes, lysis buffer, etc. It comes with a reservoir of 500 microliters, a single channel for now, and it's a very fast instrument for reagent dispensing. Another instrument that I want to highlight at the beginning is the C-Wash. The C-Wash is a new instrument that we just recently launched a few weeks ago, an instrument for liquid removal from well plates based on centrifugal forces. And the C-Wash is especially useful for automated plate washing, for lyser or other cell-based assays, but it also allows for automated bead cleanup of for DNA purification, which is very useful for NGS library prep. And with that, I want to show you one slide that comprises all our products for single-cell omics workflows, which is our today's focus. I'm really proud to say that we have now a extensive range of different instruments for single-cell dispensing, starting with the F.SIGHT benchtop instrument for dispensing of mammalian cells and fluorescent sorting. It has a single channel. The B.SIGHT, which is tailored for bacteria, and the cellenONE, which is a very flexible instrument for ultra low volume reagent dispensing and single cell dispensing. It allows you to automate entire workflows, as you will see later in the session. We then have our family of I.DOT instruments, which is the I.DOT, and we will demonstrate this later in one of the instrument demos, and the I.DOT Mini I just mentioned, and then the C-Wash for automated plate washing. I will now start with introducing you to first the F.SIGHT, which is one of our instruments of the X-SIGHT series, and the I.DOT, followed by a live demonstrations of the two instruments, and then we will have another introduction for the cellenONE by IDM. This is how the F.SIGHT looks like. It's a benchtop instrument. It allows you to insert one well plate of any format. We use a optical detection system similar to a microscope to monitor the cells inside our transparent dispensing chip, as you can see here. And by automated image processing, we make sure that only those droplets with a single cell are reaching the target substrate. As you will see later, the cellenONE is based on a somewhat similar principle. We'll talk about the details in this session. All the X-SIGHT instruments operate with a disposable cartridge, which you can see here on the slide. So how does this work? This is a schematic of the nozzle, which shows the nozzle and some cells inside. We're continuously monitoring the area around the nozzle, and if there's no cell present, the droplets are removed by vacuum suction in the X-SIGHT instruments. And if there is a single cell that matches the specified parameters, such as size or fluorescent intensity, the shutter is switched off and the cell can reach the target substrate. For each single cell that is dispensed, we generate a series of images. This is one example which document the dispensing process. As I mentioned, the X-SIGHT instruments operate with disposable cartridges. They're individually bagged and they're sterile. And it's really the only part that comes into contact with the sample, which means that there's no cleaning procedures required. There's really only a minimal setup time of typical 5 minutes to get started with a single cell dispensing experiment. One feature that I want to highlight today is the module for automated offset correction. this is something we developed to make sure that the single cells are delivered precisely to the bottom of conical wells, which is especially useful for single cell omics applications. And this camera-based system allows to make sure that cells can be lysed in very small amounts of lysis buffer. There's no manual alignment procedures required as those you might know from flow cytometry. Let me now introduce you to the I.DOT before we start to look into how it looks in the lab. The I.DOT is an instrument for highly multiplexed low volume reagent dispensing. It's based on a technology where we apply a pressure pulse to a well that has a tiny hole on the bottom to generate the droplets. And with this technology, we can generate droplets between 8 and 50 nanoliters. They can be dispensed with frequencies up to 100 Hertz, which also allows to address volumes in the microliter range very rapidly. The system can dispense into any SBS target plate, even 1536 well plates. And what makes the I.DOT really special is that it works with a 96 well source plate that allows you to load up to 96 reagents in parallel. This looks very similar to a standard well plate. However, we work with the special wells that have the tiny hole on the bottom. With this, you can dispense multiple reagents in parallel. You can basically dispense from any well of the source well plate into any well of a target well plate. I mentioned the I.DOT Mini. The I.DOT Mini has been launched recently. It's the extremely fast and affordable small brother of the I.DOT. It's the same basic technology, but here we work with single reservoirs for reagent dispensing. This slide summarizes some of the main features of the I.DOT. It's a non-contact technology, so there's no cross-contamination. It doesn't require any pipette tips. It supports multiple liquid classes. We can dispense buffers with DMSO, glycerol, or PEG, which is sometimes used in molecular biology assays, especially for NGS library preparation. It's a very flexible technology. It's one of the fastest technology out there, especially when it comes to performing complex liquid handling tasks, such as the dispensing of barcodes. It's minimal depth volume below 1 microliter, and we have a built-in drop detection, which makes sure that all droplets are ending up in the right well. Before we start to look at the instruments in the lab, which we'll do in a minute, I want to introduce you to 2 application examples, which is full-length transcript sequencing in single cells and a workflow for taxonomic classification of single bacteria. Guillaume will then later briefly touch upon single cell genome sequencing from liquid biopsies and a workflow for single cell proteomics. This is an overview of the workflow we set up for full-length transcriptome sequencing using a method called SmartSeq2. If you're interested in this, I can only refer to the talk by Simone Picelli later. He's one of the experts in this field. In fact, he is one of the inventors of this protocol, and I think there's some recent updates he will share with us today. So how did we set up this workflow? We start with a cell sample. For demonstration purposes, we work with HEK cells. We isolate the cells with the F.SIGHT using the AUC module into 384 PCR well plates with 1 microliter of lysis buffer, and then we generate the cDNA libraries and the final sequencing library. And all dispensing steps have been performed and can be performed with the I.DOT. In this case, at 10-fold reduced reaction volumes, which dramatically reduces the cost for such assays, as the reagents are the major cost driver. This is just an overview. I'm not going into detail here of how we scale down the reactions. It basically shows all the individual dispensing steps. And as you can see, we scaled all of them down by a factor of 10, starting with the lysis, the cDNA synthesis, but also the entire Illumina protocol for generating the sequencing libraries. As you can see on this slide, we derive high-quality sequencing data. In this case, we sequenced 125 cells that were prepared with this protocol. As you can see, we have a very high mapping rate of over 94%. We detect around 10,000 genes per cell at 1 million reads per cell. One thing I want to highlight, and we will see this in the video in a minute, is that the I.DOT is an ideal platform for dispensing the barcodes, for barcoding libraries, not only for single cells, but also for bulk sequencing, as it can very rapidly combine the different barcodes into 96 or 384 well plates without the need of any pipette tips. The second workflow I want to highlight is a workflow for sequencing of bacteria, in this case for the taxonomic classification of individual bacteria from heterogeneous samples. And here we use the B-site to dispense the cells into 384 well plates, and then the I.DOT Mini to miniaturize the whole genome amplification reactions in order to then sequence the cells. That's the 16S RNA by Sanger sequencing. And the challenge here is that the WGA is very costly, so we scaled down this reaction by a factor of 20, which significantly reduces the cost for such procedures. And this was possible by using the B-site with the automated offset correction and the I.DOT for low volume reagent dispensing. So here the cells were lysed in only 350 nanoliters. This is just one example of what you can get from this. This is shows the composition of phyla and cells in a human sputum sample that we analyze with this workflow. Now it's time for the lab demos. We will start with an introduction to the F.SIGHT by our field application specialist Anna Oschowitzer, and then show you a demonstration of the I.DOT system. I will now start the video. Please enjoy the instrument demo.
Hi everyone, I'm Anna. I'm field application specialist, and now I want to give you an online demo with the F.SIGHT. So I'm very happy to introduce you to the device. This is it. This is the main working space. I already plugged in an adapter for a conical 384 PCR plate, like this one here. This is the dispensing arm with the cartridge holder, and this little thing here on the left side of the carrier is the AOC module. It ensures high precision droplet positioning inside conical PCR wells, especially when you have only low volumes, so low volumes of reagents. It measures droplet positioning at the beginning of the dispensing run, so before the real dispensing run has started. And then for 384 well plate, it will measure in between 2 times, and then there's final measurement at the end of the plate, so after the whole dispensing run, to look if the droplet is still stable and is still centered like it was in the beginning. Now I'm going to show you our cartridge, how it looks like. This is it. It has a chip at the back side, and the nozzle end of this chip is imaged That's why we can really provide assurance of clonality. I already prepared a bead solution. In this case, the F.SIGHT has a blue laser integrated, so I have green fluorescent beads here. I'm gonna take out 14 microliters. You can load the cartridge with up to 80 microliters. You just put the pipette tip into this small reservoir, then fill it up with your sample. And next, the cartridge is mounted to this cartridge holder. I'm gonna mount it with 2 screws. And what I also need is an unfiltered pipette tip, which is for agitation. So nozzle clogging is prevented and your sample is resuspended inside the cartridge. I'm gonna plug this pipette tip to the adapter here, put it in the sample, swing the dispensing arm back, and put the plate on the adapter. There is a small marking, so I can't mess up the direction. And I can close the lid. So now I'm gonna share my screen that you can see how the software looks like. Let me see if the nozzle is centered. It's okay, like this. So as you can see here, I already chose a template for a 384 well plate. You can choose single wells or rows or the whole plate. I, in this case, selected the first row, and you have several actions. So you can dispense cells in bright field or in the fluorescence channel, and for single cell omics applications, you can also have a non-template control. And then you can, of course, save this template for additional experiments. And you can put in a name for the experiment, like test in this case. Here you can also use a barcode reader to have your unique barcode here for the respective plate. And here on the right side, in the corner, you can see this is a window showing you a live video. So you can see beads are coming down and entering the nozzle. And here we have fluorescence detection. So we have 2 kinds of scatter plots down here. They are used to do a pre-collection of sample information, which you need to really define your cell properties. Let's collect sample information. So you can see here, beads are coming down, and they are monitored here in the scatter plot, and you clearly see an accumulation of beads in this area. You have here roundness versus size, but you can also have the fluorescence intensity versus size. Looks pretty good. I'm gonna stop this here, and you can have magnifications of what was detected by the algorithm. So you can really have an impression about your cell type and the characteristics. and use this information for the parameters here. So in this case, our cell population has a cell size between, I think I can leave it like that, 10 and 20 micron. The roundness is definitely above 0.5. And the fluorescence intensity starts somewhere with 60 up to the maximum. So this is the first row. I'm gonna erase this one. So let's start with the dispensing run. First thing which is done is deionization. So electrical charges are removed from the plate, and then you will see the AUC measurement. Here it is called offset compensation. So the system is checking whether the droplet is really centered and really precisely deposited. And after this measurement, the real dispensing run starts, like you can see here now with the live cam, and also here. With every check mark, one single cell was dispensed, and this, or the single bead in this case, and the single bead has the criteria you set up earlier. You can also go to the experimental folder, this one here, and look at the images which were taken for every single cell dispensing event. If you have the laser switched on, you will have 15 in total. So we have 5 images for the bright field channel. This is the 4th one, and the 5th one is the empty nozzle, and you have 5 additional for the fluorescence channel, and then you have the merge of both. And of course also the values, so every other information are saved in text documents, so you can look them up and see the size and roundness and so on of your cells. So now it's performing the AUC measurement again, or it has performed, and because we received no error message, it was successful. So droplet deposition was centered and precise. Let me take out the plate. So we're now checking the position of the single beads in each well, if it's really centered or not. And I have a USB microscope next to me. And let me just switch the software, to this one. So printed in the first row here. So perfect. As you can see here, each bead is nicely centered. So as you can imagine, if you have reduced volumes of a NGS reagent, like you only have 500 nanoliters or something like that, you can really be sure that the droplet containing your single cell hit the reagent. So this was pretty much it. I think the next session will be about the I.DOT, which is a low volume liquid handler, and it's extremely useful for NGS library preparation. So I'm going to hand over to Julian and say thank you.
All right. I think thanks Anna for this real life lab demo of the F.SIGHT. I hope you could nicely see how simple it is to set up a experiment and to precisely dispense cells into PCR well plates for subsequent lysis. In this case, it was fluorescent beads to visualize them with a USB microscope. Now we will directly switch to the I.DOT demo. We already introduced the I.DOT, the instrument for highly multiplexed low volume dispensing, and I will now let our field application scientist Trang introduce the I.DOT, which will be a demo of around 10 minutes before we then continue with the cellenONE. Enjoy it.
So hi everyone, it's Chang from Dispendix, and I want to welcome you to the CellLink Partnership Conference as well. So after the introduction from Julian, I want to show you how easy it is to use the software to do your own individual protocols with the I.DOT. So what you see here is the starting screen of the I.DOT Assay Studio software, and you just have to choose which target plate format you are going to use, so which format your assay plate will be. By default, it's a 384 well plate, but you can just click on the arrow here to see all the options you have, like a 96 or 1536 well plate, or even plate formats like a 6 well plate, or custom plates that you have saved already in the I.DOT. When you're done with your choice, you can click on OK, and we'll see this overview of your plate layout. So what you basically have to do is just to click into the layout and choose which wells shall receive liquid. Those wells are highlighted in blue, and now you can decide to do different dispensing tasks, which are shown here in those buttons. So you can go for a simple transfer of liquid. That means you are just transferring a specific volume of one specific liquid into those chosen wells over here. So you go for M, choose the liquid you want to distribute over there, and choose the volume that you want to have in the wells. This is already the first thing you can do with the I.DOT, and you have more options when you want to do even more dispensing tasks, of course. So when you want to fill other wells, you can go for plus well group and choose another area of wells, for example, like those, and decide to do the next dispensing task. So now I want to do a dilution series over here. So I go for D, select a liquid that I want to dilute here as I did before. And I can decide which distribution type I want to use. By default, it's a logarithmic distribution, but when clicking into this box, you see that you have the choice of a linear distribution, or if you have custom dilution factors, you can just apply them by clicking on custom. I'll stay with a linear distribution over here, and I can choose in which volume range I want to dilute. So I will go for 500 nanoliters to 15. And according to my changes, the software will change the volumes and the steps in between according to that. Now I decide to add some replicates to my dilution series, and I want to change the dilution pattern over here. So we have all replicates shown nicely row-wise below each other. So this is basically the dilution series already. And now I want to have a backfill within my dilution series. So I go for N, which is normalization, choose a liquid I want to backfill with. So I'll take a red buffer. And now I can decide how to backfill. Either I go with the highest fluid volume, which will be the highest volume that I have within this well group, or I go with a specific volume like 1 microliter, and then my buffer will be filled up to this 1 microliter to equalize all wells within this group. So basically, I would say this is what I want to have as a assay, and I will move on with defining where to find these liquids on my source plate. So the only thing that I have to choose is the source plate type again, which is by default the standard plate already. So I click on OK. And what I can do here is either I decide for my own where I want to put the liquid. So for water, I just simply drag and drop it anywhere on the plate, or I can tell the I.DOT to assign them all automatically. The good thing here is that you always get a calculation from the system how much of each liquid you will need to be able to fulfill or perform your whole assay without any dropouts. So you have to fill in 15.2 microliters and 14.8 microliters, at least for those liquids here, to be able to dispense the whole assay. Now I'm fine with the source layout, so I move to the overview. And there I have an overview of the source plate layout, the target plate layout again, and the volumes, and can just start a simulation run here to see the order of dispensing and if everything is fine for me. I can jump in between of the steps or speed it up or slow it down as well. So when I'm happy with everything, I go to dispense, click on start dispensing, insert the plates, click on OK, and then the I.DOT will start to dispense my run. Now I click on OK again, and the result sheet will open automatically. This result sheet is generated by the drop detection feature that Julian mentioned before, which is our quality control feature, which tells you how the dispensing performance was. So if you have any dropouts, like missing liquid in any wells or not. You see green means passed, so all the liquid that was expected was also detected by the drop detection. If you have a red dot, then it means that liquid is missing. Basically, this is how you can use this software to create your own protocols already. And now we are going to dispense a dilution protocol, dilution series blue and red, which looks like this. And now we are going directly to the machine to show you how it looks like.
Now we are preparing the source plate for the dispensing run. So I'm filling the colors red and blue into the wells. And insert this source plate into the I.DOT. Now we have to insert the target plate. You see that the drawers are moving in and out automatically. Takes a bit of time. And now we insert it and go up here to start the dispensing run. Clicking on OK, and then the instrument moves in the tray by itself. Now the I.DOT is dispensing, and as you see here in the overview, all the highlighted wells are dispensed already. It will always show the progress of dispensing up here. Also, you can see the time it needs for dispensing and the progress with the bar. Right now we are dispensing buffer already, or red color in this case. And we're almost done. Now it moves out and we can see the result. Here we have our droplets just in size, so the size is going smaller, and there we have the mixture of both colors.
So, hi, it's me again. Now I want to visualize what Julian told you before. We have the barcoding protocol here for your NGS library prep. So in this case, we are just dispensing into a 96 well plate. All the S500 barcodes are dispensed row-wise, while the N700 are dispensed column-wise in this well. So we have a mixture of 2 different barcodes in each well, as you see here when you hover. And we will dispense 500 nanoliters of each of this barcode. Of course, we are able to downscale that if you want to use a higher concentrated concentrated solution of your barcodes. When going to the source layout, you see we have 20 different liquids for this protocol now, but you can easily just scale up to the whole 96 source plate. When we go for the overview here and start the simulation, you see that the S500 are dispensed in parallel, so all 8 different barcodes are dispensed at the same time, while the N700 are dispensed one by one and changing the color. The I.DOT is quite fast in doing this, so it just needs 1 minute and 11 seconds. And like it told you, it's quite easy to scale up to have really a whole plate full of different barcodes for your assay.
Okay, thank you so much, Chuang, for this nice introduction to the I.DOT. Just a reminder, if you have any questions that come up throughout the session, throughout the instrument demos, just type them in the questions panel and submit them so we can address them in the Q&A session later. Now we want to we have Guillaume introducing the cellenONE, followed by a demonstration of the system in the lab by our team in Lyon.
Thank you very much, Julian, for the intro and for the nice demo that you just did. So yes, I'm going to present you the cellenONE system. So basically what you've seen before is that we have devices to do very accurate single cell dispensing and devices to do nanoliter dispensing. And basically the cellenONE can do both these things in a single system. It's ideal because it's very flexible in terms of sample sizes that we can handle, the size of the cells we can handle, and it allows us to miniaturize a number of library preparation and to tackle some new type of methods for single cell analysis. Move to the next slide. Yeah, thank you. So today I'm just going to give you a very short overview of 3 types of applications that's going to emphasize the capability of our technology. And after that, our field application specialists have prepared a video to explain you more how the device is working. So now I will focus on the application. So we have this collaboration at the University of California, San Francisco, where they're working on multiple sclerosis patients. And what they wanted to do is to isolate specific cells from cerebrospinal fluid. The main challenge in those experiments is that they start from a fairly large volume, but the volume of cerebrospinal fluid contain only very few. So before using our device, they wanted us to convince them that we were not going to lose any of those cells, so that afterward they could undertake library preparation and sequencing on those samples. So that was a proof of concept experiment that was undertaken with our researchers there, where we tried basically to demonstrate to them the recovery rate, the fact that we are not losing any cells while doing single cell isolation. So what we did there was a set of experiments where we took a PBMC sample fairly diluted with 15 cells per microliter. We did 5 aliquots of that into a 1536 well plate using a pipette, evaluated the concentration of the sample using those 5 repeats, and then we placed a 5 microliter aliquot of that into the cellenONE and asked our device to isolate as many cells as it could. In this specific example, the average cell concentration was 68 cells. And with the cellenONE, to give you an example, with this sample, we obtained 64 single cell successfully isolated. We repeated this on a number of samples, and overall, the recovery rate that was demonstrated, or the capture efficiency, was 95%. So even with a tiny sample containing less than 100 cells, with our cellenONE technology, we are able to isolate most of the cells present. And this becomes very important when you're talking about small patient samples. Other example of application, direct library preparation plus method. That's a very nice method developed at the British Columbia Cancer Research Center in Vancouver in collaboration with the university, UBC, University of British Columbia, and Memorial Sloan Kettering Center in New York. And this is a method for whole genome sequencing of single cell in miniaturized formats. So they use a small chip where they fill thousands of single cells and then prepare the library using the cellenONE device. And this allows them to do a range of different analyses. You can do copy number inference, cluster analysis, single nucleotide variation measurement, reconstruct clonal lineages. And this is very useful when studying very specific type of cancer. And actually, this method is now in routine use at the British Columbia Cancer Research Center, where they've studied several hundred thousands of cells from patients using this method. And following the success of this method and this nice publication, a number of research centers around the world are now implementing this method. In London, we have the Francis Crick Institute that is using this method, the Japan Foundation for Cancer Research in Tokyo. Recently, the New York Genome Center just acquired a system to implement this method as well. So it's a very, very nice success story, both for the patient and for us, obviously, to see that our technology can be really used in the clinic. Finally, last couple of slides, where I'm going to present you something that we see is a really growing field in the field of single cell analysis. The ability to not anymore only measure the presence of transcripts and qualify those transcripts, but now the ability to really measure and quantify protein at the level of single cell. So what we are doing there is we are reproducing a method that is called SCOP-MS that has been developed by Nikolai Slavov. Nikolai will be one of the presenters later today and will give a talk. So I really encourage you to stay on and listen to his talk. He's a very inspiring researcher at Northeastern University. And basically what we wanted to do with the cellenONE here is a bit similar to what you do for other omics application. You isolate single cells and you have a number of preparation steps, but these are very different from your typical transcriptomic or genomic preparation. And then the analysis is not done with a sequencer here. Here we are using an LC-MS system, mass spec, in order to analyze what is the proteins that are present in your sample. And the goal of Selenium is to develop a simple, easy, fast, and reproducible platform to allow more researchers to implement these methods in order to help the field of single-cell proteomics develop. What are the steps involved in this preparation? So this is the SCOP-2-MS, the second version of the preparation developed by Nikolai at Northeastern University, as I said. In his article, Nikolai used a fax system to do single-cell isolation and then a nanoliter dispenser. What we wanted to do with cellenONE is to replace those two systems with cellenONE, basically. If you can click, Julian. Yeah, and we have set up a very nice collaboration with the IMP Center in Vienna, working with Claudia and Carl there, who are implementing these methods, and I've been having very, very nice results recently, demonstrating that the cellenONE was suitable to do both those steps. And actually, we are working on improving this method at the moment, and hopefully, beginning of next year, we are hoping to have some nice publication describing what has been made possible by Claudia by reducing the volume and even improving even further the method and making it easier to use. So that's it for me in terms of application. Just a quick roundup of the capability of the cellenONE device. It provides very high accuracy and certainty. I didn't show you the data to support that, but I invite you to look at, we have a number of presentations online that where you can see this. We can handle a very wide range of cells, and this has been really key for us and our customer in a range of applications. We are able to isolate anything from bacteria up to large mammalian cells like cardiomyocytes. As I showed you, we can also use the device as a nanoliter dispenser. We can dispense droplets from sub-nanoliter, so from about 300 picoliter droplet volume to up to microliter volumes. The platform is ideal for rare cells because we can use very, very tiny volume of cells with very few cells in there and recover most of the cells that are present. This is becoming very important for a number of different applications in the field of rare cells, such as circulating tumor cell isolation and other applications. Finally, you get actually, by being able to visualize the sample, like in the F.SIGHT, you really see what is in your sample. And by talking with researchers, we notice that this is really important for them because they can act on their sample preparation. The dissociation steps are really crucial typically when you do single cell analysis. So it's important to be able to see what's in your sample, unlike a FACS or micro droplet technologies. And finally, our technology is an open platform, so people can implement all sorts of different library preparations that they want. So that's it for me. Next, we will have a little video that has been prepared by our field application specialist dream team. So Ophélie, Flore, Sandra, Margot, and Fabiana have been contributing to the preparation of this video, and I thank them a lot for the work they've done to prepare that. Now, Julian, if you can start the little demo, and we'll be there after the video to answer your questions.
Hello everyone, thank you for joining us today. You're going to be able to discover our single cell dispensing technology, the cellenONE. The cellenONE technology is based on gentle and highly precise piezoacoustic dispensing technology. It is an automated optical monitoring of cells, and the cell's position defines whether single cell conditions are fulfilled and whether cells will be dispensed in next drop formed. So the three strengths of the cellenONE is that you can use low volumes, it has a high viability of cells, and a precision single cell isolation. So let's isolate. You can set different targets. First, you can set the target 1, target 2, or even a cellen vial can be used in the cellen vial 2 position. We usually set this target when we perform an enrichment experiment, for example. For the target 1, we can use slides, a 6 well plate, a 96 well plate, a 384 well plate, a 1536 well plate, cellen chips, or an iCell 8. For the source plate, called probe in the software, the most used plate is the 384 well plate since it has the less dead volume. Otherwise, if you want to use bigger volumes, you can use the cellen vials in the cellen wash position. So here you have your probe, your run, and target that you can choose. So today we're going to load our sample in a 384 well plate. Then we're going to use the cellenONE basic run. And the target that we're going to use is a 384 well plate as well. File, we can control the humidity and the cooling control. So the humidity you can set up as much as you want and click on set. Then you have the cooling control, so you can cool all the well plates that you have on your target and the probe as well. So here you can just turn it on here, and then you can put it at dew point or at fixed control. For example, for the well plates we use it at 4 degrees. And then you go to the field setup, and you can see the configuration we decided to do as a checkerboard. So the lighter green, you can see that we will have one HEK cell, and then with the darker green, we're gonna have one NIH cell. Then we decided to do a positive control in this one with 10 HEK cells, and a negative control in here without any HEK cell.
Once we checked our drop, we can go and load our sample. Here I'm going to aspirate 10 microliters in the E8. Once you uploaded your sample, press the test droplet button and look at the color code of the printing status light, which is displayed as a color dot in the top left corner of the PDC image. This turns green when all conditions necessary for single cell isolation are met. So here, for example, you will see the green line that represents the limit of the ejection zone and pink line that represents the limit of the safety zone. So single cell isolation parameters are met when there is one single cell in ejection zone and no cells are in the sedimentation zone. So if detected objects are surrounded by a red circle, it means that the object doesn't fulfill the isolation parameters and wouldn't be isolated. But if there is no object surrounded by green or red circle, this means that the object is not detected and therefore you would need to see the detection parameters. Once you are sure all your particles are being detected by a green or red circle, now you need to start the mapping process. So this process maps the position of cells inside the PDC during the dispensing of droplets in front of the camera. This will set the size of ejection zone that will be automatically calculated and positions according to the blue and green dots. When you see blue dots, it means that the cell detected in this location was subsequently detected again inside the PDC upon next droplet generation. Whereas green dots correspond to the last seen location of cells within the PDC. So upon next droplet generation, these cells were ejected from the PDC. Once the mapping window pops up, you'll see the computed ejection boundary separating the blue dots and the green dots detected during the mapping. Here you can see different plots, one representing the diameter of your cells, the other one the elongation factor, and the other one the circularity factor. So these different plots are going to help you to set your isolation parameters of the cells of interest. Then you can click on Set and Close. So once all the cells are being detected and we did a mapping, we can start our analysis. So here we're going to analyze 100 cells, but you can analyze more. And here, as you see, the analysis is going to be performed in the transmission channel and in the fluorescence channel. And a scatter plot as well is going to be recorded in transmission and fluorescence. Then you will have the dispensed volume and the particles detected during the analysis. A pop-up window opens once the analysis is finished. The scatter plots of different channels, transmission, blue, green, orange, and red channel will be filled with the different parameters of the cells detected. Selections can be made by holding the left click and selecting the population of interest. The number of cells that represent the selected population will then appear at the top right of every scatter plot. So here, for example, in the green channel, we have 37 cells. The displayed image correspond to the highlighted cells in green, and you can change them by selecting in the arrow. Selected cells will be displayed in green in all scatter plots. For the bottom right scatter plot, it has different axes, so intensity with the circularity, and you can change them by clicking on the gray menu on the side of the scatter plot. You can find a table with the statistics of every channel, so the transmission, blue, green, orange, and red, with the different size, intensity, elongation, and circularity of the selected cells or all the cells if no selection. With the transmission and fluorescence scatter plots obtained by the analysis, we are able to choose the optimal parameters for the isolation of our sample in transmission and fluorescence. So once the mapping and the analysis is performed, I indicate the name of the sample, so here HEK cells, and I select the green fluorescence channel in negative selection. I can check my drop with the auto drop icon, and if it is stable, I can start the run clicking on play. Here I click OK, then my target, and that is a 384 well plate, and I click OK. So here the run started, and since we are in transmission and fluorescence mode, the detection is going to follow the transmission channel first to discriminate single cell, and if there is one single cell, the fluorescence channel is going to discriminate if the cell is green positive or not. So for HEK cells, the cellenONE is going to isolate if there is one single cell in transmission mode and green negative fluorescence. This way, the printing status is green and is going to go isolate. If there is a single cell in transmission mode and this cell is green positive, the printing status is going to turn red, and therefore it is not going to isolate this cell. Then you can see two different scatter plots. The left one is the one for transmission mode, and at the right is for fluorescence mode. So you see three different colors. So the green dots are the dots that represent the cells that have been isolated. The yellow ones were the cells that were fitting the isolation parameters but were not isolated because there was another cell in the sedimentation zone, for example. And the pink dots are the cells that were not fitting the isolation parameters, so therefore they weren't isolated. So now for NIH cells, I indicate the name of the sample with NIH and select the green fluorescence channel in positive. I check my drop with the auto drop icon to see if the drop is stable and click OK, then click my target, that is a 384 well plate. So for NIH cells, the cellenONE is going to isolate if there is one single cell in transmission mode and green positive fluorescence. This way we will have a printing status in green and an isolation. So once the isolation is completed, you can set the default values as it was before, and then you can flush your PDC. And after that, you can just do the nozzle removal wash and you are done for the day. If you want to analyze your experiment after the run, you can upload your run folder and you will have this pop-up window. So here first what you can do is check your plates. So here, for example, we did a 384 well plate, but you can as well customize your target by selecting the number of rows and columns. So here you will have a checkerboard displayed as we did in the run with HEK cells. So here, if you go through this well, you will see the picture of the image of the cell that was isolated on the top right. So if you see one single cell, you can click on the well, and then with your keyboard you can put 1, so that means that is one single cell, and so on if you want to check all your wells. So once the checking is done, the overall status of the plate can be saved by clicking on this icon, and then at the end you can generate your PDF report by clicking in this icon, and then you click yes, and you will have your report generated in the folder. So here we have a PDF report where you can see all the conditions that you did the run and also the duration of the run. You have a scatter plot where you have the green dots, yellow dots, and the pink dots that were not isolated. And as well the image of your plate that you saved before. And then you have the image of every cell that was isolated. The aim of this experiment was to study the gene expression of human cells, HEK, and mouse cells, NIH. After isolation, 3' RNA-seq was performed, and results are shown in the following figures. This plot represents gene expression in HEK cells at the left and in NIH cells at the right. So as you can see, gene expression was higher in human cells than in mouse cells. This second plot compares HEK gene expression data between the cellenONE and Takara system. We can see the cellenONE is more sensitive than Takara system. And the last plot that shows that the cellenONE needs less reads to obtain more genes compared to Takara system. So thank you very much for your attention. Please do not hesitate if you have further questions or if you are interested to request a demo. And remember, life starts with a single cell.
Thank you very much, Fabiana, for this nice little demo.
Yeah, thanks a lot also from my side. Now we are almost at the end of the session, so we are now happy to address your questions. We have approximately 5-7 more minutes for Q&A session, so please feel free to submit your questions via the GoTo plugin. Let me just pull them up. Okay, we have already some questions coming in. So the way we do this now is that I will read out the questions, and then either me or Guillaume will take them. So the first one that just came in is, I think it addresses the throughput. How long does it take to isolate 96 cells with the F.SIGHT? So typically, I can take this one. Typically, you need between 4 and 5 minutes to dispense 96 cells. It depends a little bit on how much you're sorting. If you're sorting a subpopulation, This can be a bit longer, but this is what you can expect, and around 4 times longer for a 384 well plate. The second question is, what is the advantage of using the cellenONE to isolate cells compared to FACS, to fluorescent activated cell sorting? Guillaume, do you want to take this one?
Yes, sure. So there are a number of advantages versus FACS. Well, we can start from a very small population. Typically, FACS will need thousands of cells in order to start isolation. Not the case with the cellenONE. We are much more gentle. That's good for clonal outgrowth. So if you're cloning cells, wanting to let them grow afterwards, that's also very important. We can achieve higher recovery, and obviously we can do nanoliter dispensing, which a FACS obviously cannot.
Have you tried to isolate nuclei?
Yeah, absolutely. So we have a number of customers that are using the cellenONE to isolate nuclei, and they typically find that the cellenONE is more straightforward to isolate nuclei. The solution in which the nuclei are suspended tend to be a bit tricky sometimes due to the detergents that are used for preparing those nuclei, and they find that cellenONE is a very convenient method for doing that.
Yeah, same is true for the F.SIGHT. So we can detect nuclei with the systems. We would always recommend the cellenONE for applications where you have limited number of cells. As you know, Guillaume showed you data how low the loss is, and you can almost work with almost 0 cell loss. If there's plenty of nuclei, then you could go with either system. Then there is a question around scope 2, which I guess is what Guillaume introduced just before the live demo. The question is, what are the advantages of using the cellenONE compared to the kind of previous workflow? Guillaume, can you elaborate a bit more on this?
Yeah, well, obviously, like our system tend to be more economical to purchase than a FACS, and you need only one system. You don't need to buy a FACS plus a nanoliter dispenser. You can do everything with one device. One thing to note, which is very important in the preparation of the sample for scope 2, is that it's handling several organic solvents, and that's something we can also do with the cellenONE.
Okay, there's some more questions coming in, so it's not a lot, but please, now it's still the time to submit questions. We have a few more minutes, but we are also available later in the networking session. This one is probably referring to the I.DOT. Here the question is, I want to miniaturize library preparation for Illumina sequencing. Do you recommend the I.DOT or the I.DOT Mini? I can take that one. So in principle, both systems can address the same volume range. The I.DOT the I.DOT is always the better choice if you have a lot of different reagents, and this is often the case in NGS sample prep, where you typically sequence multiple samples in parallel, especially when we talk about single cells, where we want to sequence hundreds or even thousands samples in parallel. And here it makes a lot of sense to use the I.DOT, as it can dispense up to 96 barcodes at a time, and we showed you that this is sufficient to basically barcode thousands of single cell libraries. If you only are interested in dispensing, for example, a lysis buffer or a lysis buffer in the master mix, then the I.DOT Mini would be the right choice. There's another question regarding the I.DOT. Can I integrate the I.DOT onto automated workflows? Do you provide an API? Yes, we do. We actually do this for all of our instruments, including, for example, the CWash, the I.DOT instruments, and there's also ways to automate the cellenONE. With the I.DOT, it's quite straightforward. We have an API and the documentation, and both the source well plate and the target plate can be exchanged by by common plate handling robots, and so everything can be fully automated. So let me see. We had this question already. How do you make sure there's no contamination? Is the F.SIGHT sterile? That's a good question. So the instrument itself is not like, we don't have any filters. However, the instruments are small enough to just place them into common biosafety cabinets. So actually, in fact, most of our customers are operating instruments like the F.SIGHT in a laminar flow hood. When we talk about the B side, we have quite a few customers that integrated the B side into a fully anaerobic workflow, so it fits well into standard anaerobic boxes. Okay, I think we are now running out of time. Thank you very much for submitting the questions. I'm now really looking forward to the next sessions. There will be a talk by Nicola Crosetto, which will be very interesting, and then we have Ido Ahmed talking, and also Nikolai Slavlov later, amongst others. So please stay tuned. This will be an interesting day. Thanks for your attention.
Thank you, Julian. See you later.
Bye bye. Looking forward to the next sessions.